Hello and welcome to the 10th episode of the Innova Biosciences webinar series. In today's webinar, Oligonucleotide-Antibody Conjugates, an Application Guide, synthesis specialists Integrated DNA Technologies join with bioconjugation experts Innova Biosciences to explain the importance of using top-quality reagents when preparing oligonucleotide-antibody conjugates. Our first speaker today is Jessica Alexander from IDT. Ms. Alexander graduated with a BS degree in chemistry from the University of Iowa in 1997. 5 days after her graduation, she began working for IDT and has happily made oligos ever since. She oversees manufacturing operations for modified nucleic acids and GMP services. She will present the first half of the webinar, which focuses on generating high-quality oligonucleotides. Our second speaker today is Tom Speedy from Innova Biosciences. With an academic background in aquatic toxicology, Tom moved into the diagnostics industry as an R&D scientist with a UK-based forensic toxicology and drug testing company. He spent 7 years developing a number of successful products, including being part of the team that developed a saliva-based drugs of abuse test for roadside and point-of-care testing that won the Queen's Award for Innovation. Tom now manages the corporate business for Innova Biosciences, working with diagnostics, pharma, biotech R&D, and contract research companies globally. Tom will present the second half of the webinar and focus on preparing oligonucleotide antibody conjugates. And that's the basics laid out. Now please welcome Jessica.
Okay. Thanks to Innova Biosciences for inviting me to participate in this webinar. I'd like to take some time and share some helpful information about how you should order your oligonucleotides to be used with the ThunderLink conjugation kit. These oligos require a small modification, either an amino modifier for the Antibody-Oligo kit or a thiol modifier for the gold nanoparticle kit. These modifiers enable the conjugation to your molecule of interest. Also, for optimum performance, we recommend that all these oligos be HPLC purified, and I will explain why this is necessary a little bit later. But first, let me give you a little background about integrated DNA technologies. Integrated DNA Technologies is the world's largest supplier of custom nucleic acids. Each day we synthesize over 40,000 unique oligonucleotides. In the photo on the right, you can see one of our banks of synthesizers. You can see the banks of proprietary plate-based synthesizers. To enable the level of throughput that we're required to give to our customers, we designed, built, and still maintain these instruments ourselves. Not only the synthesizers, but also many other pieces of manufacturing equipment as well. Also, we have 4 manufacturing sites worldwide. In addition to our headquarters in the center of the United States, we are also manufacturing oligos in California, Singapore, and Belgium. So here's some quick advice about how to efficiently order oligonucleotides on our website. On our homepage at www.idtdna.com, click on the order box on the top banner. From this drop-down menu, one can go to a main ordering page. A screenshot's on the bottom of this slide. There's 2 paths one can choose from here. They're both marked by red arrows to order your amino or thiol modified oligonucleotides. On the left side of this screen is a shot of where one enters the sequence information that you're trying to order. It's also where you can choose your scale and your purification. Selecting HPLC purification is really critical to ensure that any residual reagents involved in the oligo manufacture are removed. If the oligo is not HPLC purified, the efficiency of the Innova conjugation will suffer a little bit. The symbols for the 3' or 5' thiol or amino modifiers are found at the bottom of this page. Good oligo design for the ThunderLink conjugation kit is really pretty simple. Here's some advice. Try to minimize the regions of homopolymer stretches. This is areas where the same base is repeated over and over. One can place the amino or thiol modification at either the 5' end of the sequence or at the 3' end. However, the 5' end is slightly favored as a position. Finally, try to avoid stretches of 4 or more consecutive G bases. 4 or more consecutive G bases can form a structure called a quadruplex, also known as a cruciform structure, and this can lower your hybridization and coupling efficiencies. It'll be useful for later discussions to briefly explain how an oligo sequence is manufactured. Let's consider a 20-mer sequence. Synthesis is going to begin at the 3' end of the sequence. Oligos are synthesized not in a solution phase, but using a solid support. So here's a photo of this support and also an electron microscope image of the support. You can see how porous it is. And each of these pores are about 1,000 angstrom in size. Often the 3' base of the sequence is covalently bound to the support. In this example, a deoxy C base is already bound to the support, and this will be the starting material for the synthesis. This support is going to be exposed to a series of chemicals in 4 different steps that's going to result in the addition of 1 nucleotide to the support-bound base. The first set of chemical reactions is delivering the penultimate 3' nucleotide to the support-bound base. In this example, it's another C base. The second step is to stabilize this newly formed bound with an oxidizing solution that creates that familiar DNA phosphodiester internucleotide linkage. Now, these first 2 steps are never quite 100% efficient. There's always a small percent, usually less than 1%, of the growing polymer that does not couple. Therefore, this third step, this step will cap any of those unreacted species as to prevent the presence of any deletions in your final sequence. The fourth and final step in the synthesis cycle is unmasking the growth protecting group on that newly added base. Now your oligo is ready for the next base addition. These 4 steps making up the synthesis cycle only require between 3 and 8 minutes, depending on the scale of the synthesis. And repeating all of those steps again will add the 3rd base to the oligo. Once more, the 3rd cycle will make an oligonucleotide that's 4 bases long. Then repeating the cycle for a total of 19 times will give you your complete 20-base sequence. This can be done in as fast as an hour. The synthesis cycle is almost 100% efficient with each addition, but never absolutely perfect. The efficiency of this cycle is referred to as the coupling efficiency, and it can vary with reagents, instruments, and especially between oligo vendors. IDT recognizes that a small decrease in this coupling efficiency can result in significant differences in the amount of target species that are in your oligo. It's the most important metric that we monitor in all our manufacturing systems. This slide is a graph showing the relationship between oligo length and the percent full-length product. IDT maintains a coupling efficiency of 99.25% across all our locations and scales. If we consider an example like that 20-mer, a small loss in coupling efficiency will decrease that final purity by about 5 or 10%. But this difference becomes really profound for longer oligos, and if we can consider, say, a 75-mer, you can see a 1% decrease in coupling efficiency can result in a loss of purity in your crude product of almost 20%. But even when your coupling efficiency is very robust, it's still necessary to purify your oligonucleotides to achieve purities greater than 80 or 90%. We recommend HPLC purification for these amino and thio modified oligos. So here is an example of a 75-mer oligonucleotide that's modified at the 3' end with an amino group. Before purification, as measured in this capillary electrophoresis trace, you can see that there is only 62.7% of the target sequence. And you can see all of those truncated sequences eluting earlier than the full-length product that comes out at 18 minutes. So everything earlier than that is sequences where there were small deletions in the sequence. After HPLC purification, and this was a really good purification, we were able to achieve 98.4% purity. To conjugate your oligo to your molecule or particle of interest, it's necessary to label the oligo with a nucleophile, such as an amino modifier or a thiol modifier. And here is the structures of two of our common 5' modifications. You can see the functional group is at the end of a 6-carbon linker. Let me show you why it becomes important to further purify these oligos that have been modified with these groups. I sometimes like to think of these groups as kind of a molecular dust mop. They do tend to pick up whatever might be in their proximity rather indiscriminately. The very reason why these are such useful functional groups that enables them to react and attach to the molecules that we want them to also makes them a little tricky to manufacture because we need to ensure that we're not allowing these very reactive sites to react prematurely and become blocked and not useful. Here is an example of a 30 nucleotide sequence with a 5' amino modifier group. And this is an analytical reverse phase trace on the left before HPLC purification. It's 70.8% pure. And if we look a little bit more closely at around the full-length product, we can see the presence of another species that's eluting later than the full-length product. This is the blocked amino modifier, and it's present in this lot of oligo at 8.8%. And that's a fairly significant amount. Now here is that same lot of oligo that's being analyzed by a different technique. This is a really neat analytical tool at IDT, electrospray ionization mass spec. Every oligo at IDT is analyzed by this technique, and it can tell us really useful information. One might consider it to be kind of a molecular ruler. It will tell us the size of the molecule. But in addition to giving us this qualitative information, it also tells us if there are other species, other masses present in the sample at a significant amount. And here we can see clearly the presence of two heavier masses indicating some blocked amino modifier. With HPLC purification, though, that peak is fairly readily removed. So on the left is our lot before HPLC purification with some blocked material at 70.8% pure, but after HPLC purification you can see that that process readily removes that blocked species, giving an overall purity of 94.4%. IDT has validated manufacturing methods to try to minimize the formation of most of those unwanted reactants, but for any that might form, the proper purification will ensure the highest purity oligos. So I'd like to summarize by saying that good oligo quality is achieved by maintaining robust coupling efficiency, and using HPLC purification will also ensure that your oligos are of the highest purity. And now I will give control over to Tom. Thank you.
Thank you, Jess. That was really interesting. I'd like to lead on now from where Jess has left off with the synthesis of the oligos and go into a little bit on conjugation of oligonucleotides to antibodies and the applications that these conjugates are being used for. Firstly, I'd like to give you a quick introduction to Innova Biosciences. We're just over 10 years old. Founded in Cambridge in the UK, originally providing assay services to the pharmaceutical industry. And then in 2005, we launched our custom antibody labeling technologies. The four main product categories that we have are the Lightning Link, that is for labeling enzymes, fluorescent dyes, biotins, fluorescent proteins and such to antibodies. We have the Innova Coat range for nanoparticle conjugation, currently covalent coupling of gold nanoparticles to antibodies or other proteins for the detection of various targets through lateral flow and flow-through assays. Most recently, we've launched the ThunderLink technology, which we'll be covering today, for the conjugation of oligonucleotides to antibodies, proteins, golds. We've been offering this as a service for a number of years now, but more recently we've launched the technology as an easy-to-use kit for do-it-yourself labeling. And our other product range is the phosphate detection kits, which are used in the pharmaceutical industry for high-throughput screening and such. And all of these technologies are covered in other webinars available at the Innova website. So traditional immunoassays, two main formats, direct or sandwich. In the direct assay, for example, competitive assays, one detection antibody is used to detect the target. In the sandwich, you have a capture antibody and a detection antibody. These antibodies need to be labeled in some way to be able to visualize the detection. For this purpose, enzymes such as HRP or alkaline phosphatase can be used, or fluorescent dyes or nanoparticles. Here are a couple of examples. The traditional ELISA plate using HRP with TMB substrate, and the lateral flow assays with gold nanoparticles, here showing a competitive assay with a positive result and a negative result. More commonly, we see the pregnancy test in the sandwich format, where this would be the positive and this would be the negative. So traditional immunoassays, colorimetric detection, they tend to have a microgram to picogram detection limit. We need a relatively small amount of sample material in the microliter region, but this sometimes, in some cases, could be quite large if only very, very tiny amounts of sample are available. They can be used to detect a variety of proteins or haptens or other antigens, and it gives a linear signal with endpoint detection. It's a very well established technology. It's been around for many years now, can be used on a huge number of different types of sample matrices, and can tolerate lots of different contaminants in the sample. So improving on this, we need to find a way to overcome the limitations of colorimetric detection, sometimes background issues using colorimetric methods. And to this end, in '92, Sano and his team proposed the oligonucleotide labeled immunosorbent assay. The detection limits of these are in the picogram to femtogram region, so 100 to 1,000 times more sensitive than the ELISA, and can work with much, much lower sample volumes. So they combine the specificity of the antibodies and all the benefits of traditional immunoassay technology, but they harness the power of the amplification available within qPCR, giving us an exponential signal and real-time detection. This high sensitivity, it requires higher quality reagents. I'm sure all of you that work with different assay formats have found that when you have relatively poor or contaminated start material, the quality of the assay and the sensitivity is much harder to achieve. These immunosorbent oligo-linked assays also tolerate a variety of matrices and contaminants. So quality and sensitivity is directly related to the reagent quality. This requires high quality oligonucleotides, such as those Jess was describing and those available from IDT. The conjugate must not contain fragments of oligonucleotide, and it must also not contain unbound oligonucleotide material. Some oligo-immunoassays can tolerate unbound oligo material, but on the whole it causes high background and basically invalidates any of the benefits that we would have from that increased sensitivity through the high background that you have when amplifying non-specific oligo material in the sample. So traditional techniques for conjugating antibodies to oligo are very costly, time-consuming, and really you require expert knowledge to be able to do this. Lots of different methods have been tried. They tend to be unreliable and often result in cross-linking or free oligo material in the conjugate. Some people have tried to overcome these difficulties by using biotinylated antibodies and then streptavidin labeled with oligo, but again you then risk the cross-linking and background associated with the use of streptavidin in some assay formats. So what we've done at Innova is to develop the ThunderLink technology to overcome these problems, and as I say, for a number of years we've been providing this as a custom service to a variety of partners. Some of the technologies I'll present a little bit later demonstrate these uses and these applications, but most recently we've also launched a kit for people to be able to do this conjugation in their own laboratories. The basic principles are very similar to our other conjugation technologies, the Lightning Link or the Innova Cope, where you take an amine modified oligo and then couple this to the antibody through our proprietary chemistry. We also include positive controls to ensure that you can check that the conjugation has worked properly, and it's a unidirectional conjugation, so you won't have antibody-antibody or oligo-oligo formations in your final conjugate. We also provide an unbound oligonucleotide removal stage within the kit so that you can eliminate a significant amount of any unbound material, and we offer this as an enhanced service within our custom services as well. So the basic principle is the activation of your antibody, the activation of the oligonucleotide, a cleanup of this, mix the two together, and then you have your antibody-oligo conjugate. A very reproducible system, very easy to do in your own laboratories, and doesn't require a significant amount of expert knowledge beyond basic laboratory skills. Here we can see some of the gels that we get when we do custom conjugation for the QC to determine that the conjugation has been effective. You can see here in this non-reducing gel that we have the antibody here, and then when we have the conjugation you can see that it shifts on the gel, and you get this variation based on the number of oligos bound to the particular antibody. In monoclonals you can actually see a banding occurring within the gel. And this tells us that all of this antibody has now been coupled to the oligonucleotide. On a reducing gel, you can actually see the light chain and the heavy chain and the oligo conjugate to that on the DNA gel shows us where the oligo has actually bound to those heavy and light chains. In the reducing gel, because of the way in which that conjugate is formed, not all the chains will be labeled with an oligo because you're fragmenting the antibody, and therefore only some of those will show up as labeled on the DNA gel. So the ThunderLink technology, it's compatible with oligos between 10 and 120 bases. We have had customers who've been successful at larger than this, but as Jess mentioned, that it's not possible to synthesize high quality oligos beyond this unless you move into next generation sequencing, which we're not going to cover today. The aiming group can be at the 5' or the 3'. We include positive controls, and all the necessary components are supplied within that kit. If you're actually developing a commercial assay and you feel that some form of optimization would be beneficial, or if you're interested in our custom optimization services for tailoring this conjugation specifically to your requirements, then please do contact us and we can provide you with more information on that. So what do we use these oligo-antibody conjugates for? Why are we bothering with this conjugation process? As I mentioned earlier, there's the immuno-pcr assays. I'll discuss a little bit more about that in a moment. We have proximity ligation assays, proximity extension assays, and then a unique application of the oligo conjugation technology in 3DNA technology for lateral flow and for cellular targeting that I'll cover towards the end. So combining these immunodetection and nucleic acid amplification methods, we have a variety of different areas that this has been applied in toxin pathogen detection, cytokine multiplexing, pharmacokinetic studies, molecular interaction analysis, huge number of different areas that this is now being applied. Here we can see a graph showing the number of occurrences of immuno-PCR related publications over the last 10 years. And as you can see, there's been a steep rise over recent years as we've overcome many of the problems associated with the traditional methods, the fragments, the high background, the difficulties associated with coupling. As we've overcome these, you can see that the application of this and the number of people working with these oligo-antibody conjugates is increasing significantly. So this is a forefront area of science at the moment for people working in diagnostics and lots of other areas. So immuno-PCR, Sana and his team back in 1992 published this paper in Science, and they gave an example of the replacement of that colorimetric enzyme-based assay technology with the combination of real-time PCR to provide a massively enhanced sensitivity in the standard immunoassay format with the coupling of an oligo to an antibody. A really nice example of this technology being applied is in the plague paradigm, paleomicrobiology investigation, where the degradation of DNA over time meant that it's not a very reliable way of them determining the contamination of teeth samples from graves. PCR was struggling because of this degradation of DNA. So what these guys did was to actually do a comparison between traditional ELISA methods, PCR methods, and the immuno-PCR method. They found that the protein concentrations that could be detected was 70 times lower using immuno-PCR compared to standard ELISA, being able to detect 0.74 nanograms as opposed to the 50 cutoff used in ELISA. They used 34 teeth from mass graves of plague victims, and 10 negative teeth were included. The ELISA had detected 3 of the 34 positives and had 1 false positive. PCR achieved 10 out of 34, and immuno-PCR 14 out of 34. The limitations on this were partially due to the cutoff that they set, but as you can see, the immuno-PCR was able to improve on the traditional methods that were being used. And there are lots of other areas where this can be applied to provide us with an enhanced sensitivity over traditional protein immunoassay technologies, coupling that with PCR and overcoming some of the problems of DNA degradation that limit the use of PCR in some applications. Another application is proximity ligation. Here we can see Olink have developed a unique system whereby they use 2 antibodies to detect a particular target, and then when these are detecting and they come into close proximity, the DNA strands interact and they use rolling circle amplification to generate several hundred-fold replication of that DNA, which is then labeled using a complementary oligo probe, resulting in a really high concentration of fluorescence in each single molecule amplification. So you get a really bright dot when this is viewed under a fluorescent microscope. And you can see here these oligo probes catching on this proximity ligation here. A further development on this principle is the Olink proximity extension assay, using the ProSeq probes with 2 oligos labeled. When they bind to the target, They then are complementary, so they extend, and then this is then detected through the real-time PCR. Another representation of this is the mixing of your antibodies labeled with oligos and the sample, causing this ligation via the connector, amplification through PCR, and you can see the different targets here being detected via different oligo combinations, resulting in a multiplex analysis and quantitation by real-time PCR. So traditional multiplex assays often suffer from cross-reactivity. What Olink have done is to overcome this and to expand on the opportunity for multiplexing through the use of different complementary oligos. So 1A will only bind with 1B, 2A, 2B, and so on and so on. And therefore, these unique DNA oligo sequences will only report where they're matched in pairs. So you don't have these same cross-reactivity problems. For more information, contact us and we'll put you in touch with Olink, or certainly find more information on their website. So a unique application of our oligo conjugation technology is in lateral flow, where Genosphere have developed this 3DNA technology. They've applied this to lateral flow to enhance the sensitivity of lateral flow tests. In doing this, they've taken the antibody, the traditional immunological detection reagent, coupled this with an oligo to their 3-DNAmer, which has multiple golds bound to that DNAmer, giving a huge amplification in the signal through this aggregation of gold on the dendrimer. In comparison to a traditional sort of streptavidin biotin gold system, you can see here that the intensity of the signal will be much reduced through using this system. This is a good example of a technology that really does require no unbound oligo material to ensure that you're not blocking off the binding sites for the antibody on the dendrimer, and therefore you get the maximum number of antibodies, or the desired number of antibodies binding, to ensure that you're getting an optimal conjugate forming. And the final application of this technology is the Genosphere DNA technology in cellular staining. And here you can see a fluorescent 3-DNA customized with a transferrin antibody coupled to an oligo, looking at HepG2 cells. You can see here with the Cy3 DNA, Cy3 3-DNA, there is no antibody and you're getting no background on this, so no staining at all. When this is then coupled to the antibody, you can see here a really clear and bright staining of that same cell using that antibody-oligonucleotide conjugate. So here, I hope this has been a useful and interesting start point in the discussion of oligonucleotide antibody conjugates, a fascinating area that's really starting to pick up and gain huge interest through the elimination of the problems initially associated with the quality of the reagents, both in their independent forms of oligo or antibody, but also in the complexities and difficulties associated with actually bringing these together, conjugating them efficiently and reliably, and their application in a variety of different areas. For more information, certainly send us an email, have a look on our websites, feel free to give us a call, and we'll be more than happy to discuss your requirements. So thank you ever so much for your attention, and I'll hand you back over to Claudina. Thank you.
Thank you very much for joining us today. I hope that you enjoyed the webinar and that you'll be able to join us for future webinars in 2014.